My name is Daniel Tward. I'm an assistant professor here at UCLA in the Department of Computational Medicine and also the Brain Mapping Center. Today I'm going to be talking about ways of comparing spatial transcriptomics data sets by embedding them in a metric space, so we can compute a distance between any two data sets. And in doing so, I'm going to present a mathematical model which can describe spatial transcriptomics data sets at many different spatial scales, from the micron scale all the way up to the centimeter scale of tissues. So let me start with a bit of a review of what spatial transcriptomics is, and MERFISH in particular, which is what I'll be focusing on today. MERFISH stands for Multiplex Error-Robust Fluorescence in situ Hybridization, and I'm going to not say that again for the rest of the talk, so I'll just use the acronym MERFISH. Now, spatial transcriptomics is a really exciting family of technologies. 2 years ago, in 2020, it was crowned by Nature Methods as the method of the year, and I've extracted a couple figures from this report. In the top row, we can sort of see the multi-scale nature of these data sets. Can you see my mouse? Yep. Over on the left-hand side is a coronal section of a mouse's brain. You can see some very small fields of view where imaging data was captured. At every pixel in the imaging data, we're able to sequence RNA, which gets expressed, and identify locations in this image where a particular RNA molecule was expressed. So the idea here is that fluorescent probes allow localization of RNA. A sequence of images allows sequencing of these RNA probes. So the probes bind to the first nucleotide, a chemical reaction occurs, they'll then bind to the second nucleotide, and they'll glow in a different color depending on which base pair is expressed. After doing this a few times, we can map these fragments of RNA to individual genes. And in the panel over here, you can see the result of this mapping, where we see what genes are being expressed at what locations in space. Now, this type of data is naturally hierarchical. We go from single molecules, and as we zoom out, we move to cells and to tissues and to organs. So on the bottom left here, you can see an example of how these RNA particles are grouped into individual cells. And a group of RNA molecules inside a cell can be used to classify what type of cell that is. Continuing to zoom out, a group of cells of different types can be used to classify what type of tissue this is. And so this data has a very natural way of being interpreted at many different spatial scales, with many interesting and potentially nonlinear transformations between the scales. So here's an example on the left-hand side of a MERFISH dataset, part of a MERFISH dataset. What you see here is just the locations, XY coordinates of every cell which was detected in a coronal section of a mouse brain. So I know most people in this audience aren't particularly familiar with neuroanatomy, so I just wanted to give you a little bit of a walkthrough of what the image you're looking at is, and introduce the idea of a brain atlas. So on the right-hand side here is one slide from the Allen Reference Atlas. This was built at the Allen Institute by Hongwei Dong, who is another faculty at UCLA, and some of his collaborators a few years ago. And so some of the important features you can notice here as you look at these different colored regions, which are annotations, is that on the outside of the image, we see the cerebral cortex of the mouse. At the top, there's the motor cortex, which drives how muscles move. In the next block, there's the somatosensory cortex, which begins to process inputs for the sense of touch. Deeper to some of those structures in green, you see the hippocampus, which is important for learning and memory and really stands out in a lot of these images on the left-hand side. Below the hippocampus is the thalamus, which controls all of the inputs to the cortex. To the right of the thalamus is the basal ganglia, which is responsible for more complex motor planning. And at the bottom of the image here, in red, is the hypothalamus, which is sort of the link between the brain and the endocrine system. Now, what's interesting is you can see the way that these brain regions are subdivided into smaller and smaller compartments. And you can't really see those subdivisions when you look on the left-hand side or even the left-hand side of this atlas. So neuroscientists use very complex patterns of cells, their layering patterns, knowledge of what connects to what in order to annotate this data set on the right. It has a lot of information in it beyond what you can tell just by looking at something on the left. Now we can zoom in on this little red box and see the level of single cells. So on the left-hand side, we see something that looks a lot like a Nissl stained image. The purple image we see over here is a Nissl stain. As you zoom in, you can resolve individual cells as opposed to looking at something which seems almost like a cell density. Now in this data set, there's 85,958 cells that were detected. And for each of those cells, we have a sequence of measurements associated with how much RNA was expressed in that cell for each gene. So here's an example shown in green of the expression of a gene called HTR1B. And you can see that this gene has a very interesting spatial pattern. It's expressed very strongly in the basal ganglia, but it's also expressed in the hypothalamus and throughout the cortex. I'll show another example of a gene called HTR1F, and this one you can see is predominantly expressed in the cerebral cortex, but in the inner layers rather than in the outer layers. And there are some genes like this one, OXGR1, that are expressed throughout the brain, and they don't have a particularly well-defined spatial pattern. Now, at this point in time, spatial transcriptomics technologies are fairly new. And so my goal in working with this technology, rather than to answer very specific hypotheses, is just to map out what are the cell types in the brain, how much variability there is in cell types, and where are all these cell types located. Now, because this data is so new, the community hasn't really agreed on what sort of model is appropriate for looking at this data mathematically, or how the data should be stored computationally. The community isn't really sure yet how to answer questions like, are there individual differences in the spatial distribution of gene expression? And if so, at what location? And one of the important facets to questions like that are, how can we even identify the same location in different individuals? Now, what makes that latter question so difficult comes back to the hierarchy of this data from very fine spatial scales out to very coarse spatial scales. So at the finest level, different people or different mice have different cells. I can't compare neuron number 100 in your brain to neuron 100 in my brain because we don't have the same sets of neurons. If you are a worm, that may not be true, but if you are a mammal or a human, that's definitely true. However, at the coarse scale, as you zoom out, individuals, other than in the cases of pathology, have the same sets of tissues and the same sets of tissue compartments. And as we transition between these two resolutions from the very fine scales to the very coarse scales, we move to something called the mesoscopic scale. And this is a term which was put out by my colleague Partha Mitra a few years ago to kind of speak about at what scale it's meaningful to compare neurons in the brain. So we are proposing a specific model for describing data sets like these, and I'm going to talk a little bit about the mathematics of this model called a varifold. So I'm going to spend a little bit of time explaining what a varifold is, and then I'm going to talk about how we can use it to compute distances or dissimilarities between spatial transcriptomics data set. So a varifold was proposed in the 70s and 80s, around that time, as part of geometric measure theory. So it was first used to describe things like surfaces that are created by bubbles. And if you look at the surfaces on this book, you can see all sorts of very, very weird geometries, from normal surfaces like a cylinder, to circles that have noodles coming out of them, to Mobius strips, all sorts of things like that. So this is a very generic object, which is useful to describe complex geometry. And it's also used to describe features. In the case of this book, the features are normal vectors and tangent vectors. But in the case of spatial transcriptomics, the features are gene expression profiles. So let me get a little bit into the mathematical details. Varifolds model particles, like lists of cells with their location, as well as images, like regular grids of pixels, in a common framework. So this mathematical tool allows us to pass from the microscale of RNA particles to cells to images seamlessly. And varifolds account naturally for variability at the microscale, and I'll get into what exactly that means. So the fact that we don't have individual cells that correspond to one another, but rather we have neighborhoods such that the average cells within that neighborhood are similar between me and you. So in terms of notation, we have a list of 86,000 cells. One of them, called cell I, is located at location xi, and brings with it a list of features, fi. We have, I think, about 150 genes that were sequenced in each cell. And so the varifold, which models the distribution of cells and their gene expression throughout the brain, is denoted by here the Greek letter mu, which is a mixture of little empirical distributions of every cell. So I'll describe exactly what this notation means. This delta xi is like a little spike that encodes the fact that the cell is located at xi. And this delta fi is like a little spike that encodes the fact that we observed the features fi. So in this model, the w's are weights in this mixture. And as I mentioned, xi can be arbitrary locations, like lists of cells, or it can be regular arrays, like images. So I know this is, a lot of the work in genetics is discrete, so people who are not so familiar with these symbols, delta functions, I'm going to do a little bit of a refresher. So these are called delta Dirac measures, and if you refer to them as functions, certain mathematicians will start yelling at you, but it's okay to call them functions from my point of view if you want. The idea is we consider a probability density function like a Gaussian, which you can see one of them here in yellow. And we allow the width of that Gaussian to start shrinking and shrinking. So as the width of the Gaussian moves from yellow to green to light blue to dark blue to purple, its width starts shrinking and shrinking, and its height grows and grows and grows. And in fact, as the variance of this Gaussian, the standard deviation sigma, goes down to 0, the height of this function keeps growing to infinity. So this sequence of functions does not have a limit in the usual sense. But it does have a limit in what's called a weak sense. So one thing you can do with probability distributions is use them to calculate expected values of certain functions. So over here on the left side, we have a blue function, f of x, and an orange function, g of x. And we can calculate the expected value of these functions with respect to the Gaussian distributions as the width of the Gaussian starts shrinking down and down to infinity. And what you can see is that they converge to some constant value. They converge to, in fact, the value of the function at the point where this Gaussian distribution is centered. So while these infinitely thin Gaussian distributions, which are called Dirac measures, while they're not well defined in the sense of functions, they are well defined in the sense of how they act on things in order to give expected values. That being said, this operation is only well defined as long as the functions that they act on are nice. So the functions should be smooth, continuous. They shouldn't oscillate infinitely quickly. They shouldn't go unbounded up to infinity. If they do, then we won't be able to have this nice property where we can use infinitely thin Gaussians to really measure a random variable that has no uncertainty to it. So this is like an empirical probability density function. So as I mentioned, we can consider how probability densities act on functions to give expected values. So the notation I'm using here, this closed circle, means the action of p on g, where p is a probability density, g is some function. We can write it as this integral over x, or we could just denote it as an expected value with respect to the probability distribution p over the function g. And this action between probability densities and these functions is a linear action. So probability densities can be thought of as linear functionals, which are maps from functions to the real numbers. So varifolds, up to a normalization, varifolds don't need to have an area under them equal to 1. Varifolds up to a normalization behave in the same way. If we have a function g, which is a function of space and features, we can act on them with our varifold and create a real number, which tells us the action of that varifold on a particular function. So we can think of these varifolds as linear functionals, which are dual to a space of nice functions. So these are functions which don't oscillate infinitely quickly, don't go to infinity, anything like that. Now, because this action on functions is so critical for thinking about varifolds, we need to have a model for what it means to be a nice function. And to do that, we use a tool called reproducing kernel Hilbert spaces. So this is a tool which is used very commonly in curve fitting or data fitting with non-parametric models. So it's a way to get smooth, nice fits through your data without assuming a particular form of the function. And all these nice functions are just weighted linear combinations of kernels, k. And in our case, we'll always use Gaussian functions as kernels. So we can describe any nice, well-behaved function as a sum over Gaussians of some bandwidth. So as an example, down here, we show a varifold as a whole bunch of points, and the color of the points represents some features attached to them. So 3 features are attached here, which respond to red and green and blue values. And what's nice about reproducing kernel Hilbert spaces is that there's a duality between these varifolds and the smooth functions that they can act on. So for every varifold, we can produce a corresponding smooth function and vice versa. So for this varifold you see here, if we act on it by convolving it with a smooth kernel, we get a smooth function like you see here. So this is a tool for converting between these very, very ugly, infinitely tall, infinitely thin empirical distributions, converting them into nice, smooth functions, which can be sampled in any way we like, and doing it in a way which is invertible. So we can pass back and forth between these varifolds and these smooth functions using tools available in reproducing kernel Hilbert spaces. Now, what's nice about this framework is if we model our spatial transcriptomics data as a varifold, we can compute the norm of this varifold because it's just a vector in a Hilbert space. So in terms of the variables we've defined, the norm of this particular varifold is a double sum over the weights in the mixture, a Gaussian kernel evaluated at pairs of cells, and the dot product between features. So what this means intuitively is the norm of a varifold is small if nearby features are not similar to each other, and it's small if all the cells are far away from each other. So unfortunately, calculating this norm is quadratic in complexity. So you see it sums over I and it sums over j. And I and j in this data set are both 86,000. So this ends up being very complex. And if this is something we want to do optimization with respect to, it ends up very, very complex. And if we want to be in line with the goals of the Brain Initiative Cell Census Network, for example, all these technologies are now scaling from mice brains up to primates and human brains. So it would really be hopeless to work with a model that requires quadratic complexity. But there's a trick associated with reproducing kernel Hilbert spaces that exactly the same norm can be calculated by converting this varifold halfway into its dual function. So if we use a Gaussian kernel with half the variance, we can convert our varifold into a smooth but not so smooth function. And then the norm of the varifold is just the sum of squares of this function, the integral sum of squares. So this representation is useful mathematically because it allows us to look at neighborhoods of cells, where a neighborhood is defined by the width of this Gaussian kernel K. And it's very useful computationally because it allows us to reduce an n squared complexity calculation down to an order n complexity calculation. Okay, so this is just a summary. Generally, in a vector space, if you have a norm on some vector, you can also compute the distance between a pair of vectors. So if I have two spatial transcriptomics datasets, mu and nu, I can calculate the distance between them in a very standard way using these kernels K and the dot product between features. And the intuition, like I just stated, is the two varifolds are similar if their densities and features are similar when averaged over some neighborhood. And the previous quadratic complexity function that I mentioned to you now expands into three different quadratic complexity functions. So when we're looking to compare data sets, the complexity grows even further. However, if we use this dual representation that I mentioned, once again, we can just look at sum of square error between these dual functions I and j. All right, so we now have a tool that can be used to compare two spatial transcriptomics datasets by asking if they're similar when averaged over small neighborhoods. But we also need a model to account for the multiple scales at which this data lives. And so what we've done is built a hierarchy by alternating between spatial aggregation, so grouping particles together into cells, grouping cells together into voxels, alternating between spatial aggregation and nonlinear transformations of the feature vectors. So we move from RNA particles to genes to tissue types. These are nonlinear feature transformations. So moving from left to right, we start with RNA sequences at various points in space in panel A. We then group them together, we aggregate them into cells, and we transform the features using, in the example I'm going to show you, we just do PCA, and then we apply k-means to the first few components. There's a lot more complex and interesting ways to do that, but I wanted to show something simple here. So we convert the RNA expression profiles into cell type profiles. Then we can aggregate over large pixels, which you see in panel D, and once again do PCA and k-means to convert these mixtures of cell types into tissue types. So let me just show you an example of what that looks like on our data set. On the left-hand side are all our 86,000 cells. We do PCA to extract a few components that account for most of the variability, and k-means to divide it into 20 cell types. So on the left-hand side, every cell type has a color. The RGB value of the color is equal to the first three principal components of the gene expression profiles. Then on the right side, so in step E, where we convert from cell types over a list of cells to tissue types over a regular array of pixels, once again, we have about 20 tissue types. And what you can see here is something that looks very similar to the annotated brain atlas that I showed on slide 2. So many of the brain regions that I described show up clearly as a tissue type here. Over here, you can see the hippocampus and some of its subfields. You can see the cortex in bright red, as well as some of its layers and parcellations. You can see a strong difference between the basal ganglia and the thalamus, for example. So we're really classifying brain compartments in this manner. Now we can compare data sets just by summing norms across scales. So we have a varifold at each scale. We'll collect them all together and we ask, or we state, two data sets are similar if their varifold representation is similar at the RNA scale and at the cell scale and at the tissue scale. All right, so when we compare two data sets to each other, and we want to use the idea that locally distributions of cells and their features are similar, we need to be comparing locations in space that correspond to the same part of the brain. So if we have two data sets over here, and I want to apply the varifold norm to this image on the left minus the image on the right, if I want to know how similar they are in the hippocampus, this has the coordinate x equals 4,000, y equals 5,000. I can't just go to the right and look at the coordinate x equals 4,000, y equals 5,000 in order to make a comparison, because that's comparing apples to oranges. It's comparing the hippocampus to the thalamus. So before we can make meaningful comparisons about the difference between these data sets, we need to be able to align them accurately. So the approach to do this is to lay out a coordinate grid on one of our images. You can see we have a grid set up on the left, and find some sort of transformation which will move all the points on the grid, carrying the image with it, such that we've transformed the shape of the image on the left to match the location, orientation, and shape of the image on the right. So this is an example of how we can do it by stretching out this coordinate system. If we can do this, we can establish a corresponding coordinate system between pairs of spatial transcriptomics data sets, and we can align them and make comparisons between their varifolds to see how similar or different they are as a function of genes or as a function of space. On the other hand, if we use a standard coordinate system, just like you often align genes to a standard reference genome, we have a way of comparing any data set to any other data set. They just need to go through this standard coordinate system. And in neuroscience, the Allen Reference Atlas is such a coordinate system. Yeah, you had a question?
Yeah, exactly. So maybe you referred to that in your last question. So my question is, probably you can distort the coordinates such that you will be able to align everything to everything. So what is this what you said?
Yes. Well, I want to be careful about what you mean by everything to everything. So we're going to restrict ourselves to a family of transformations that preserve topology. So we won't introduce any tears or holes, and that leaves neighborhoods intact. So the fact that certain regions are always next to certain other regions, that'll be preserved under these transformations.
But the transformation is over the coordinates, not over the figure.
That's correct. So I'll show toward the end of the presentation some examples where we also apply transformation to the features, and we can use that to align different types of spatial transcriptomics data sets. So I'll show an example where we compare gene expression in MERFISH to gene expression in Visium. I think there was a talk earlier in this conference about Visium. Does that satisfy your curiosity for now? When we get to that point, you can ask me some follow-up questions if you want. All right, so the framework we use to model and compute these types of transformations was first established in 2005. It's called the Large Deformation Diffeomorphic Metric Mapping Framework. This is a framework for constructing spatial transformations that align data sets such that they preserve topology, and that they are always invertible, and such that they're parametrized in a way that we can actually compute them, and we can assign distances to them. So we can quantify how similar are two data sets in terms of their shape, as well as quantifying how similar they are in terms of gene expression. So I'm going to quickly go over this diagram on the right. Usually when I give a talk, I spend half an hour talking about this. And today I'm only going to spend 1 minute talking about it, so that means you guys are lucky. The idea here is if we have two imaging data sets at the bottom left, this is a surface that describes the hippocampus in the brain of one image. At the bottom right is the hippocampus of the brain in another image. To go about aligning them, we'll first lay out a time axis. Here, time is parameterized between 0 and 1. We'll then construct a smooth velocity field as a function of time. You can see that in the second row. And then we integrate this velocity field over time to get a smooth displacement field, which I show in the third row. So as you know, the integral of velocity over time gives displacement, and we're just doing this at many points in space altogether. When we have these displacement fields that you see in the second last row, the displacement fields can act on our imaging data to deform them, and we'll compare how similar this deformed blue surface is to this red surface, and conduct an optimization procedure in order to make them as similar as possible within limits that we are preserving some sort of regularity. And R stands for regularity, is equal to the distance between the identity transformation and this transformation squared. So when we compute transformations, we make sure that they're meaningful and they respect biological organization by introducing regularization that makes sure they are not too big and that they are smooth and invertible. So over the past year or so, we've been building a tool for aligning data sets, spatial transcriptomics data sets. There's one issue that needs to be identified before we can actually use this tool, and this points to another nice feature of varifolds as a model for our data, which is that when we transform spatial coordinates, this is going to change cell density. And so normally, if we compare a big person to a small person, in a big person, it is not the case that their cells are further apart. It's the case, generally speaking, that they have more cells. So if you look on the left-hand side, our data set got transformed. There's certain regions where it's squeezing and the cell density increases, and there's certain regions where it's expanding and the cell density decreases. And so if we don't transform these data sets carefully, we'll end up with an image like the one you see in the middle. And I've highlighted two regions where the data is expanding, and so the density goes down. And on the right-hand side, the data is compressing, and so the density goes up. So in fact, varifolds come with a tool for describing how they change under change of coordinates, and they change by adjusting their weights based on the determinant of the derivative of this transformation. So what this means is if the cells are stretching further apart, their density needs to go up. And if the cells are compressing closer together, their density needs to go down. And this is sort of a correction factor that tells us by how much they need to go up or down. So we can build a dissimilarity function in order to align two data sets. I'm calling this function M for matching. And our dissimilarity function includes the varifold norm between the dual images. So we compute these nice, smooth images by acting on our data sets with a kernel. We look at the difference between them and compute sum of square error. Only after inputting this function phi, which allows us to transform features of one data set to match the features of another data set if necessary. And adding a weighting term here, W, which tells us that there might be some regions of the image that need to be ignored. For example, if there was missing tissue, or if there was corrupted data, or if there was artifacts. So using this matching function, we can construct an alignment algorithm. And I'm working with a colleague named Jean Fan at Johns Hopkins, and she has a lot better branding than me. So this is called ST-Align instead of EM-LDDMM plus spatial transcriptomics varifolds. So I think ST-Align is a nice word, but I'm not particularly good at branding, so I need other people to help me with that. So we simply align two data sets by optimizing a cost function. We use an expectation maximization algorithm to minimize that. We want to make the two spatial transcriptomics data sets as similar as possible by minimizing this matching function. But at the same time, we don't want to make them similar in such a way that we estimate transformations which destroy biological organization. So we include this regularization term, which corresponds to the size of the coordinate transforms. Now, the expectation maximization algorithm is a tool for getting maximum likelihood estimates when some of your data is missing or corrupted. So in our algorithm, we first conduct an E step where you estimate this function W. So we estimate where data might be missing. That's done by Gaussian mixture modeling. Then we compute an M step where we optimize over the parameters in our shape change and the parameters in our feature transformation. And we alternate these two steps until convergence. The EM algorithm was first published in 1977. There are some arguments that it was developed a long time before that and maybe Dempster, Laird, and Rubin don't deserve all the credit, but they still deserve a lot of credit. So I'm going to show you some results aligning pairs of data sets and validating accuracy by computing in small regions how gene expression is correlated between two replicates. So on the left side here is a spatial transcriptomics data set. And on the right side is a transformation of a different spatial transcriptomics data set with the goal of spatially matching them so we can compare gene expression profiles. So in the 3 rows in this data set, we are comparing gene expression profiles of 3 different genes. And what we did is break these images up into small chunks. And each chunk, we look at what is the gene expression in one image and what is the gene expression in the aligned image. And we look at the correlation or cosine similarity between them. So in this gene, GABBR2, we see they're fairly strongly correlated, and the cosine similarity is 0.9. In this gene, GPR6, we see the correlation is a little bit weaker, but they have a cosine similarity of 0.8. And on the bottom, a third example, we look at this gene CCKAR, and we see a lower cosine similarity of only about 0.6. So to my knowledge, this is the first time looking at how reproducible these types of technologies are. So these replicates are different mice brain sliced at approximately the same location, imaged using a particular MERFISH pipeline, and we're just trying to compare before and after, one replicate versus the other replicate. Are the gene expression profiles we measure the same? Yeah?
So it seems like the diffeomorphism, the alignment is actually using the expression to align, right? So if that's the case, then wouldn't you expect after alignment the correlation to be high, to be inflated?
That's an interesting question. We would expect it to be high, but that's the signal that we use to align things. So we definitely expect it to be higher than chance. I don't believe that this is somehow, I don't believe here that we're cheating essentially. And let me explain why. These data sets were aligned using a much simpler signal. So we weren't using all of the genes to align the data sets. In fact, here we were only using cell densities. But I think it would be fair to make a comparison like this if we hold out a few genes, don't use them to drive the alignment, and then do use them to evaluate the accuracy. But I think you raise a really good point, which is that in this field, we still don't know the best way to validate in terms of accuracy or reproducibility. Yeah? Yeah. So what a pseudospot is, and I didn't come up with the word pseudospot, is this sort of small region in the brain that we've extracted. And it wasn't extracted on a regular grid. It was extracted using some sort of k-means procedure. But basically, it's just some small region in the image. And we'll look at both of our data sets restricted to that region, evaluate the total expression of a certain gene within that spot, and every spot becomes one point on this scatter plot. In terms of the units here, so the units are how much a particular gene was expressed in that region, and the units are only valid up to a normalization constant. So we can look at their ordering, and we can look at their relative size, but they're not particularly meaningful on their own. Different technologies will have different sensitivities. Different runs, even of a MERFISH technology, will have different sensitivities, so we may get more or less expression, but the relative expression between different genes could still be the same. Does that answer your question? Okay, great. So the next example we looked at was comparing two different technologies. Oh, yeah?
Yeah, sorry, one more question. So let's say if the target is missing a piece compared to the source.
Oh, perfect timing.
Okay, yeah, I've seen that. So would the diffeomorphism try to stretch and fill in that missing piece, or would it not even bother to align that missing piece?
So there's a trade-off, and the trade-off is really determined by this parameter W. So if we can do a good job estimating W and estimating where there is missing tissue or corrupted tissue, we won't stretch things out to match them in those regions. And in this case of the MERFISH to Visium match, because our Visium signal was only approximately a hemisphere, we estimate where there is good data present that we can drive the alignment, and we can estimate where there's not good data present to avoid driving the alignment. And so you can see here we can align a whole brain to a half a brain by as accurately as possible estimating where there is missing data that should be ignored when we compute a change of coordinates. So when we compare gene expression for the same 3 genes using these 2 different technologies, using the same measure as I just described to Harold, we end up with similarity numbers that are a lot lower. So here there's, for the gene BAIAP2, the cosine similarity is only 0.8. For SLC17A6, it's 0.7. For GRPR151, it's 0.6. So this is really, it should serve as a warning for how mature these technologies are. If we measure the same thing with 2 different technologies, we might get different numbers. If we measure the same thing even with the same technology, we might still get different numbers. So the third situation we're applying this alignment tool is to align our 2D coronal cross-section data sets into a 3D atlas so that we could use all of the labeling that was constructed in the first page to see if there really is an assortment of cell types into the regions which were invented by anatomists. So the partitions that I showed you on the second slide, there is not a good sense that the brain knows it's supposed to be partitioned in that way. Those were invented because they were convenient or because there was a particular type of signal that someone was looking at such that they thought there was a partition. So now that we can map these 2-dimensional data sets into the 3D atlas, in addition to estimating deformations, we also have to estimate the angle and the anterior-posterior coordinate where they're located. We can now compare gene expression within every compartment defined in the atlas. And the Allen atlas defines approximately 1,000 different compartments. All right, so let me finish up. I think I'm going to be right on time. I just wanted to summarize. MERFISH provides gene expression profiles for every cell in a brain image. It's a very promising new technology that can be used to catalog all of the cells in the brain. We are proposing a model for analyzing these data sets called varifolds, which is a tool that came out of geometric measure theory, and analyzing them at multiple scales by alternating between spatial aggregation and feature transforms to extract meaningful signals at each spatial scale. We can align these data sets efficiently by understanding the images that are dual to these varifolds. So we can overcome quadratic complexity by applying some tricks that are known in the field of reproducing kernel Hilbert spaces. We can compute these alignments between images of the same modality, between different modalities, or even between 3D and 2D. And we're starting to apply these tools to validate the reproducibility, the accuracy of these new technologies, as well as cataloging all the cells in the brain. Now, the next interesting steps for this type of computational analysis is not just to say, well, where are all the cells and the cell types, but to ask specific hypotheses, to say, if somebody has a disease, where does their gene expression differ between healthy individuals and diseased individuals, and which genes does it differ? So we're beginning to develop tools to answer those questions, but what I presented on today was mostly tools for cataloging all the cell types by minimizing dissimilarities between data sets. Let me just thank some people. Osagie Emi Uwu is my student from UCLA Big Summer who's been helping with spatial transcriptomics data set analysis. Jean Fan, Kaylin Clifton, and Michael Miller are colleagues at Johns Hopkins University. And Alain Trouvé is my mathematician colleague from Paris. This work is funded by the BACCN, Brain Initiative Cell Census Network, and Jean Fan is funded by NSF Career. And the data we showed today came from Jean Fan and her colleagues from Harvard University. So thanks very much for your attention. I'm happy to answer any more questions if there are any.